This video is going to go over the basics of bacterial transcription and the coupling of transcription with translation in bacterial cells. Let's really quickly review what we call the central dogma of molecular biology. Remember that DNA is used as the template for the process of transcription, which results in the formation of a messenger RNA. Those mRNA transcripts then get translated into protein. And all proteins have some sort of functions. They can be enzymes that carry out catabolic or anabolic processes, or they can be structural proteins that provide shape to a cell, like actin in our cells or MREB in bacilli. You all remember that RNA is made by an enzyme called RNA polymerase. RNA polymerase is a DNA-dependent RNA polymerase, which means that it uses DNA as a template and makes an RNA molecule. The bacterial RNA polymerase is what we call a core enzyme, and it's composed of 5 subunits: beta, beta prime, alpha double prime, and omega. Now this core enzyme doesn't associate very tightly with DNA, so as it scans along the DNA it has trouble staying on and it can just fall off. And it also has trouble recognizing promoters of genes. So this little core enzyme of the bacterial RNA polymerase needs some help to do its job really well. And that help comes in the form of another subunit called the sigma factor. When the RNA polymerase core enzyme associates with a sigma factor, it forms what's called the holoenzyme. Now that all of the pieces are there and in place, the RNA polymerase is ready to transcribe some messenger RNAs. Here we have a diagram of just a generic RNA polymerase, and the polymerase unwinds the DNA as it moves down that strand, and the DNA closes up behind it, so there's no sort of bubble like we see in DNA replication. As the DNA threads through the RNA polymerase, it's the bottom strand that's used as a template, and the newly transcribed messenger RNA just sort of hangs there from the end of the polymerase by itself. Imagine these 2 black lines represent the promoter of a particular bacterial gene. This area is called the +1, and it's actually the transcriptional start site, or the site at which the RNA polymerase will begin making a messenger RNA. Upstream of, meaning ahead of, the transcriptional start site is an area called the -10 element. This region of the promoter has a particular and conserved sequence that goes TATA, and for that reason we often call it the TATA box. And the final promoter area is further upstream. It has its own sequence as well, but it's simply called the -35 element. Together, all 3 of these regions make up the promoter for any given gene, and all 3 are necessary for the RNA polymerase to bind and for transcription to proceed through the gene. Okay, now let's take a zoomed out look at what's happening. Here in red is that entire promoter region, and now let's draw a little polymerase and add a sigma factor. So our holoenzyme complex is going to bind to the promoter region, and because that sigma factor is present, it's going to bind tightly and it's going to stay on the DNA. And now our polymerase is ready to begin transcription at that +1 transcriptional start site. The RNA polymerase begins moving down our DNA strand into the gene, and it will start by making a tiny little transcript like this. However, that tiny little transcript will dissociate from the RNA polymerase. This is termed abortive transcription because it doesn't go anywhere and it ends very quickly, and it can happen multiple times at the beginning of each round of gene transcription. So the polymerase goes back to the beginning, reads a little bit, makes a short transcript, and continues doing this because the way forward is blocked by sigma factor. You see, what needs to happen is that our RNA polymerase moves forward, makes that tiny little transcript, and then sigma factor must be released from the holoenzyme so that only the core enzyme can move forward. And as it moves forward, it's going to continue elongating that RNA strand until we have a full messenger RNA, and transcription of our gene proceeds until we reach the termination sequences within the DNA coding sequence. Now, bacterial species can have many different sigma factors, and each of those sigma factors can have their own set of functions. Let's look at E. coli, for example. E. coli has 7 different sigma factors, and the most used one is sigma 70, which is the sigma factor responsible for the transcription of what we call housekeeping genes, or the basic genes that control metabolism, growth, and all of those various necessary functions. Sigma 19 is involved in iron transport into the E. coli cell. Sigma 24 regulates genes that are necessary for responses to extreme heat stress. Sigma 28 regulates the genes involved in flagellar motility. Sigma 32 is another heat shock sigma factor. Sigma 38 is active in transcribing genes when cells are in a starvation mode, and it's important in cells that are in the stationary phase of a bacterial growth curve. And the final E. coli sigma factor is sigma factor 54, which is activated during times of nitrogen limitation. One more time, sigma factors control the expression of genes and the differential expression of genes by binding to the RNA polymerase core enzyme, forming the RNA polymerase holoenzyme, and binding it to the appropriate promoters to allow some genes to be expressed and other genes to not be expressed. For eukaryotic genes, each gene is under the control of its own promoter. However, for bacteria, that's not the case. Here we have a piece of bacterial DNA that has 3 different genes: our blue gene A, the yellow gene B, and the red gene C. And all of these genes are going to be under the control of the same promoter, which I'm going to shade in green. When transcription proceeds through all 3 of these genes, the result is a messenger RNA that has a 5' cap and a 3' poly A tail. Only this messenger RNA is what we call polycistronic. So one cistron is one gene. So this polycistronic messenger RNA includes the information to make the protein encoded by gene A, and the protein encoded by gene B, and the protein encoded by gene C, all within one single message. And now this transcript is ready to be used for translation. Because bacterial cells don't have a nucleus, there's no need for the attachment of nuclear export proteins to our 5' cap. And interestingly enough, bacterial transcripts don't undergo splicing. Now, the last thing to cover in this video is that bacterial transcription and translation are coupled. And that means they can occur simultaneously. We don't have to wait until transcription of a messenger RNA is complete to begin translating that messenger RNA into a protein. Why can this happen? Well, remember, bacteria don't have a nucleus, so we don't need to export our messenger RNAs from the nucleus to the cytoplasm. Because transcription is occurring in the cytoplasm, those messenger RNAs are accessible to ribosomes immediately after transcription begins. One of the major benefits of this to very small organisms like bacteria in ever-changing environments is that it allows them to very, very quickly change their gene expression in order to adapt to the environment they find themselves in. Here we have an RNA polymerase that has already transcribed most of a gene, and you can see the messenger RNA hanging from that RNA polymerase in the blue line, and it already has its 5' methylguanine cap. Now let's add some ribosomes to our messenger RNA. So our pink ribosome binds to that 5' methylguanine cap and starts to move up the messenger RNA, adding peptides as it goes. So this is what it's meant by the coupling of transcription and translation. Now, not just one ribosome can bind to a messenger RNA. In fact, many ribosomes can fit on the same transcript at the same time. And this allows for a greater level of protein production than if only one ribosome bound to each transcript. So here on the bottom I have a messenger RNA, and I'm drawing a whole bunch of little pink ribosomes. As soon as one ribosome has moved far enough down the messenger RNA, another ribosome can bind and begin translation itself. And the result is a messenger RNA bound by multiple ribosomes, each of which is at some part of the message making a protein. And these messenger RNAs that are laden down with ribosomes are called polysomes or polyribosomes because there are many ribosomes bound to that one message. This is really great for small organisms like bacteria because it means they can use one single messenger RNA to produce lots of proteins very, very quickly.